All right, welcome everyone. We see the attendees streaming in. We are now live on Facebook. Sorry for the technical glitch there. We're trying to do this ourselves. As I said, John will not be with us this week or next week. We have Karen and Terry with us, who will be managing the Q&A and the chat, as well as any questions that are coming to us from Facebook Live. This week, we will be covering basically a clinical recap so that we can get questions on a diverse array of topics, as well as we'll focus initially on convalescent plasma, which has been in the news. And quite a few people have been asking questions throughout the week to me about convalescent plasma and what is the actual controversy about the FDA speeding up its approval under emergency use authorization. So I'll cover what it is, what it can do, and what are the current questions from the medical and scientific community about its authorization. Then we'll take any, as we go along here, as usual, direct any questions to us via the Q&A panel, or that's where we prefer the questions to come in so that we can manage them and collate them. Commentary in between between attendees and the panel can happen, of course, as usual, as it does in the chat. And of course, FB is a good place for questions as well if you're not joining us on the Zoom. Okay, so what I'm going to do is I need John or Terry to give me the ability to share screen.
I am not the host, so I cannot do anything right now.
Terry, do you want to give me co-host or something like that?
Yeah, I'm working on that. John, we're going to miss you horribly.
It's not as easy as it looks.
We're doing this on purpose so John feels valued.
John, you're so valued. All right, so
Oh, good. I'm good.
You got it now?
Yes. So let's go ahead and start this slideshow. Okay, so as we discussed, we'll be going over a recap clinically of therapeutics, current state of vaccinations, because that's a lot of areas where there's quite a bit of news coming through. As I said, convalescent plasma is in the news this week, with yesterday the FDA speeding up its emergency use authorization. The oddity of that is that even without that authorization, convalescent plasma has been broadly used throughout the United States. And it has been spearheaded by those who started a group called Survivor Corps, which is which now consists of thousands upon thousands of people who had coronavirus, COVID-19, recovered, and then shared their stories with each other, which allowed us to gather a lot of the information on the course of the disease, symptomatology, and they're now sharing also their plasma. And that plasma has been used throughout the country, but most recently the Mayo Clinic published some of their results that indicated that across a broad spectrum of patients that were monitored who had received convalescent plasma, about 37% of them fared very, much better than their expectations based on their clinical picture. Now, what does that mean? That's what we're all struggling with in the scientific and biomedical community to truly understand. This is not the result of, this 37% number is not the result of a randomized controlled trial with, some patients receiving, not receiving plasma versus those receiving plasma, otherwise known as placebo, for those who are not. So we really still have to truly understand. That's why there's a lot of pushback from those in the CDC, NIH, and private, university researchers urging the FDA to be cautious about this emergency use authorization. The I'll discuss that, and you know, what are the things that we're worried about not having data on. Vaccinations, I've been getting a lot of questions on the timeline for availability. With, you know, the news is full of announcements from either companies saying that we've entered this phase or that phase, or we've got, you know, 3,000 or 10,000 people now receiving their phase 3 of the vaccination. There's news from the UK, which currently is out in front, that we'll discuss a little bit about. We'll discuss what is safe and effective. One thing that we will, that I want you to take note of on the screen here, and we'll have to discuss further and get input from you guys about where you are and what you're seeing with your numbers. This precipitous drop, if you can see over here, in percentage, well, in number of cases per 100,000 that are being reported. This is problematic because this isn't how a pandemic behaves. These numbers clearly are not accurate. So my kid, who has been, you know, exemplary about gathering data from various sources and putting it together and explaining under each graph his observations and analysis, I post everything that he posts to our resource page. And what's very clear is that I won't go so far as to say the data is being manipulated, but the data is not clean. It's not directly from the source of information. And therefore, there is some sort of, you know, there's, I'll call it systemic inaccuracy that we're seeing in all of these numbers. This is not how pandemic behaves. You don't have a curve that, you know, rises like this and then suddenly drops off. There is conjecture, which I probably support, that these are numbers that are in states that are politically important for the election. And you know, that's disturbing, because that won't give people in those regions and counties and states an accurate risk assessment. And that's bad for public health for lots of reasons, which, you know, if you have any questions about, we can talk about it. Curves are dropping overall. Percentage positivity is dropping. Whether that is reliable information is still uncertain. I'll just leave it there. What to beware of in the fall? All indications are that the coalescence of the flu season and potentially a true second wave of COVID could be quite disastrous. There is some good news that the primary prevention methods that have been continually stressed by us for the last 6 months, and now more vehemently by those who suffered this outbreak in the South. Masks work, distancing works, not engaging in large gatherings works. And if you really needed to go through that hell to be aware of that fact, well, there you have it. So we lost thousands of people. We overloaded ICUs, and hospital systems were at their breaking point throughout the South, and some still continue to be. And that is a direct result of the ignoring of this public health messaging. And if anyone wants to talk freely about how this prolonged the pandemic, anyone I want to engage in that discussion, we'll do that towards the end of the session today, because I think it's important for not just me, Terry, and Karen, who've been stressing this, but to get your perspective on how it has hurt you as, you know, individuals who expected, as we did, to kind of be out of the woods by June, and here we are mid-August, and we're not. And what has that done to your family, your health, your career, your community? I think those stories need to get out there and be heard and be powerful, because if they are not heeded, then we'll be sitting here in October, as Terry has said, October or November, and staring at these curves going back up. And I don't want to do that. Okay, so I'm going to mute for a second and try to get things quiet. Terry, if you want to make any commentary, be right back.
Oh, I was going to see if John would come on and dance. He usually jumps in and dances at this point, but I know John's had a bad week and he's got a trip to get ready for. So go ahead, Sanjay.
Can you guys hear me? Okay, good. I'm back. All right, so let's go over a quick comparison, because this is where we want to start off. The awareness of what is the real not the real, but the estimated numbers of key metrics here. With the flu, the R naught is about 1.3. Case fatality, depending on the severity of the season, can be anywhere between 0.5 to 2.1%. And COVID has proven much, much more deadly. That's very evident. The incubation time is quite quick for flu, right? You know, if you get exposed, you start to feel sick within 1 to 4 days. The typical symptoms of fever, body ache, are very evident quickly. The comparison with COVID is that can take much longer. You can get exposed. 40% of those who are exposed will not show symptoms, and they'll just get to a point where they're able to spread. So 4 days later, and up to from between 4 and 10 days, they can continue to spread to others without ever feeling, quote unquote, sick. The hospitalization rate for flu on the high side is 2%. Generally, the elderly and those with chronic respiratory diseases suffer the most with flu. Community attack rate is basically what percent of a geography will be affected in the event of an outbreak of flu. So for about let's say a community of 100,000 people, 10 to 20%. All right, so about 10 to 20,000 people will get the flu during that specified amount of time of what we regard as a season in that specified geography. Annually around the globe, about 1 billion people are infected. United States, about 10%, I would say, less than that, maybe 7 to 10% on a bad season, a bad year, could get infected with flu. And then we have anywhere between 10 to 61,000 are the estimates of flu deaths a year. And again, the elderly are affected, but so are the young. We don't have data on, obviously this is an ongoing and novel virus. We don't have data on how many people are going to be infected this year. We have estimates. We're already at 24 million globally, and about 5.7 million here in the United States have been infected. But as I said, those are, 5.7 is a proxy for about 9 times more than that actually having suffered the disease and never been tested positive.
Sanjay, do you use the John
Yes.
Hopkins site for data? Okay.
Yes. It's been the most reliable. It doesn't suffer from data hiccups, and it's normalized using their own public health algorithms. So, it's from 6 months ago till today, we know that it is underestimating the count. They make it clear that the count is underestimated and that those numbers are proxies, but they don't attempt to scrub the data. It's just as it is. And we've been able to use that for lots of, good purposes. So it's really not necessary to talk about SARS and MERS, but I wanted to compare flu to COVID for people to have perspective. Are there any questions at this point on anything with these numbers?
Well, I have a question from Chai. She thought there's a lot of people that have been evacuated for the wildfires in California, and they are in shelters. And is that a worry, having people be in shelters?
Very much so. Very much so. And in fact, last week we talked to a woman from, I think, South Central California, who mentioned that the extreme temperatures it caused the local public health people to set up cooling tents, and even that can cause people to congregate just to cool off, especially the homeless, and that becomes a nidus of infection. So
I think there was actually Chai last week we talked to.
Yeah, but there was someone else that had joined us. Anyway, the shelters should be providing masks. I think Californians know by now that that this remains a risk to their health to even be among people in a gathering of more than 10 people from different households. In this crisis situation is terrible. It's very difficult to manage. But with the use of masks, even in that scenario, we should be able to reduce transmission. Okay. All right. So one of the things that we had covered, I think, a couple of weeks ago is the advent of rapid testing. We unfortunately now, this is week 6 of the resurgence now coming down, but throughout this period of time, people have reported that the wait times for results extended all the way out to about 16 days. And on the short end, we're about 3 days. So, and again, we've been stressing the importance of knowing the results within 24 hours, if not sooner. So we as a nation need to transition away from the swab test to the rapid antigen test. The PCR-based swab test to this rapid antigen testing method, so that this becomes ubiquitous and that you can get your results in 5 to 15 minutes. Unfortunately, as recently as this week, I have heard from people in Illinois, Florida, Ohio, that their test results have extended beyond 5 days. So that's not good. And again, I don't like sharing anecdotal evidence, because I get people contacting me when they're scared and when things are not going well. And I wish I could report that 90% of everyone else are getting their test results back in 2 days and things are going swimmingly, but I can't, because I haven't heard those stories. So I'm not saying those stories are not out there, that we're not being, we're not doing better than we were 6 weeks ago. I would hope that we are, and we continue that trend, but this is where we need to get to. Karen, Terry, and I, and John have talked about how that if we're going to get through this pandemic sooner rather than later, it's the primary prevention that is going to carry us, but it's going to be the testing that helps us end it. Okay. So what I mean by primary prevention, it's carrying us right now with mask usage, not gathering, not having sporting events, and it sucks, right? I don't like going shopping like this. I don't like not having football. All of that. No one is sitting at home going, "Oh yeah, this is awesome," unless you're really weird, and that's fine. Weird is okay. I'm weird too. But the thing is, we can't we can't expect this to go away. It's not going to disappear. We need to find out where it is, and that means testing and isolation, testing and tracking and tracing. If we don't know where it is, it's going to just constantly keep cropping back up. All right. The irresponsibility of having teachers and children of all ages flood back into campuses without testing all of them and having a strategy to have them tested every 4 days at minimum, and having the technology available to do so, is a massive national failure. There's no other way to put it. Okay. You know, I love Terry, and sometimes his cynicism is highly merited. Like when he said, you know, people rush out and paint graffiti on a courthouse, and you know, you got 1,000 people wearing $10,000 worth of gear there in 24 hours to try and disperse them. But you have, you know, 8-year-olds flooding back into crowded schools, and nobody is helping them figure out, you know, if they're coming in from a home that has been exposed, if they're going back out to a home that has elderly in them. Are their teachers carrying the virus right now? How do you create a bubble around these schools to keep kids and teachers safe? You know, I think the cynicism about where we pay attention and disperse our resources, the cynicism is merited. Okay. And so these are the machines. Now, I have a great report from Mark Cameron, my friend down in Florida, that now he, you know, who is a commercial pilot and, you know, is doing the highly responsible thing of taking on his own testing, when his own airline and his fellow employees of that airline have not been offered a testing regimen to keep themselves and their passengers safe. Why? Because it hasn't been federally mandated. The FAA has not come forward and said, this is what you must do. The CDC has not come forward and said, this is what airlines and airports must do. There is no leadership on this, but my friend, Mark, who is a commercial pilot, is getting tested regularly because he has found a place in Jacksonville, Florida, that has this test over here, the Quidel Sophia 2 rapid test. And he can get his results in just a couple hours. Why is this not everywhere? Okay, that's a major issue. I don't know if we have to make noise. Well, we'll start making noise. I mean, because this can't continue. And if we don't do something about it, we will see a massive second wave in October, when all of us are living indoors and forced to live or operate more of our lives indoors, where we know the spread is more likely. Okay, so this seasonal variation that we saw, punctuated by a outbreak that took place across the warmest states in our rocket. No, sorry guys. You know, across the warmest states, when we were told, oh, during the summer there'll be a lull, is a prime indication that weather doesn't matter to the virus. Our weather dependent behavior matters to the virus. Okay, so once we're indoors because it's cold and miserable out, the virus is going to be very, very happy to spread. And if we don't have testing and other resources, and we're not vigilant, then we will see a second outbreak. So that's what I wanted to cover here. Anyway, so questions so far?
Thank you for saying my cynicism was merited, because
You're welcome, because it was.
I got more where that comes from.
Oh, I know. So here's the next step is, this is a bit of a stopgap, which is stuff that you can buy off the shelf in your drugstores, right? So, you know, this is a slide that indicates that eventually we'll have strip-based testing, right, of the antibodies, which means in your recovery phase, you'll be able to do a quick pinprick like you do for your blood glucose, and you'll be able to see, all right, well, I'm 6 weeks out off of recovery, IgG has shown up, which means that I have some measure of protection for the next 2 to 3 months minimum, and maybe even up to a year until the next season. This doesn't mean that we don't suggest and we don't urge you to get a vaccine once the vaccine's available. If you have recovered from COVID and you test positive for antibodies, and you don't have to go through the whole going to a lab like Terry did, giving blood and then waiting and all that stuff. This is a test that you could potentially buy in the near future off of the shelf at your local drugstore. You know, a quick pinprick, put it on the paper, and then add the thing, and you know, within a few minutes it tells you if you are positive for the antibodies. Anyway, so this will be available. The previous test will be available hopefully very shortly, and we'll start moving towards moving away from waiting around for test results, and we will be able to do a lot of this kind of stuff at home. This holds true for not just the antibody test, but the antigen. Does everybody understand the difference? Yeah? Okay. So here's this is just the previous slide. This slide is just expanded into the process that happens where you add the reagent, add your blood, blah, 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 go through it. Could be blood, it could be, yeah, I think it will be blood in this case because we're looking for antibodies. And then the test results will show up here, okay? A quick, you've seen this slide many times. I've shown it to you. I'm happy to say that we were among the first to gather evidence that doctors across the country or health teams across the country were utilizing these therapies. Now, I want to take a moment and make very, very clear. We do not have anything that cures COVID, okay? We don't have anything that you can take to prevent you from getting COVID. What's Karen holding up? Oh, yes. And yes, thank you. But what we have discovered that has, wow, rocket, man. Okay, so what we do have is numbers that have shown that we have cut mortality, which is the in-hospital deaths of patients that are positive for COVID. We've cut mortality down from about 2.5% down to 0.8%. And it's dropping even lower. We should see it drop below 0.5% by the end of the summer. And that's very impressive, okay? That's very impressive. That's cutting mortality down by a fifth. In May, I said by the end of June, we'll be down by a half. And now we're mid-August and we've cut mortality down by a fifth, by 4/5. So 4/5. So this is impressive stuff, and it's because we've used our knowledge of how the virus attacks the body to stop it from doing the worst damage to the body. And that is that it goes after the it activates a cascade that causes microclots throughout the body, okay? And these microclots have been responsible for strokes, for severe cardiac damage, and severe organ damage. When we figured that out in about May, we started using low molecular weight heparin to prevent damage in these patients, and the success rate has been enormous. And there'll be studies out talking about how, you take 23,000 patients and you look at those who fared poorly without this treatment and those who fared well with this treatment. And then, we can really start to understand how impactful it was. But just clinically, now we know what its direct effect is and how many lives it saved. We've also done the same with giving immune modulators. And these immune modulators are methylprednisone, dexamethasone, and those are standard steroids that we give to calm the immune system down when it is in overdrive. And one of the things that COVID does is it kicks the immune system into overdrive. It defeats interleukin-6 and the cascades that that result in a natural antiviral response. And so in order to stimulate that, we can actually give immune modulators like interferon beta and others that will then recalibrate the immune system to prevent the harm that it does to itself, and also to promote the natural antiviral effects. Okay, so that's what we know. Now, convalescent plasma. What is it? Well, you have a COVID patient, they recover, you draw blood, there will be virus neutralizing antibodies in their blood. You purify down the serum plasma, and then you can give it to doctors and nurses that are treating COVID patients, and that is prophylactic, and I'll explain why it's prophylactic on the next slide. Or you can give it to patients who are sick. The studies show that the patients that come in with the initial deterioration symptoms, that means they're having difficulty breathing and they have a intractable fever, okay? But they're not at the stage of the cytokine storm and the shutdown of systems, and you need to prone them and vent them, and not that stage. I'm talking about they've been sick for 4 days with intractable fever, and now they're having difficulty breathing, and they come to the hospital. At that stage, when you give them convalescent plasma, it seems that they do very well, okay? And even in some patients that are really at the edge of death, as a last-ditch resort, about a third of those patients respond to convalescent plasma. Now, everybody in between, we still don't have enough data. We don't know whether it's effective or not. Now, why? What is going on here? Well, it's very complicated, but think of it this way. Not everybody who gets their blood drawn who has recovered from COVID will have the same level of neutralizing antibodies, okay? And right now, and we talked about this over 3 months ago when this first came up. You can't dilute the plasma. So you can't get a bag and give it to 10 patients. It has to be a one-to-one therapy, okay? So one patient receives it from one donor. And the reason for that is titration and all this other stuff, and the duration of which they have to receive the therapy. We won't get into the details of dosage and all that, but just think of it this way. Let's say all of us on the panel, Terry, myself, Karen, and John, all get COVID and recover, okay? Maybe my recovery was 4 days, but Karen's was 12. Maybe I got a very mild case and Karen actually suffered through fever and cough and all of that stuff, okay? The measure of your sickness is also going to be somewhat correlated to the measure of your neutralizing antibodies. And it's directly proportional. So the sicker you got, the more powerful your immune response and a higher titer or concentration of neutralizing antibodies will be able to get out of your plasma. If you had a mild case, then more than likely all of the really complex stuff that ends up activating T cells and the B cell response, that didn't kick into full fifth gear there. It just kind of helped, but your innate response and other factors, including the fact that you had a much lower viral inoculum, means that the concentration of your antibodies for those recovering from a mild case is going to be much lower than Karen's who had a worse case, okay? Now, we actually completely ignore that right now. So we just pool all of that all of the patients as one characteristic: they recovered. We don't say, okay, here's the group of patients that recovered from severe disease, moderate disease, and mild disease. We just have plasma. And now when you give that to a patient, you're not matching them with their case status. So I'm not giving mild plasma recovery to a mild plasma patient. I could, in fact, be giving a mild plasma recovery patient donor to a severe patient. And therefore, I will not be having a maximal effect with that patient. Do you understand? So this is why the FDA said, wait. We don't want to really approve this yet because the safety characteristics are not all fleshed out, and it's not fair to all patients to say that this is going to have a 37% reduction across the board, because we don't know that. In these 19 patients that were studied, the results were remarkable. Okay, but let's go back and find out who they got those antibodies from. What were the actual characteristics of the antibodies they got? Okay, and then you need to kind of dig deeper and dive down into effectiveness and all that stuff, because they were not only that, there were different dosage regimes. Okay, some were 200, 300, and 400 ml in one dose. Some were 900 ml in 3 doses over 7 days. Some were 2,400 ml in 8 doses over a month. Anyone want to guess which one's the most effective? Which one's the one the FDA is pushing? Okay, this is the problem with pushing a therapeutic out and glossing over the details. This is the problem with pushing remdesivir and under emergency use. Now, their argument is that people have a right to try, people are dying, and at this point, we're trying to save as many lives as possible. And to that, I must say I largely agree, okay? But the counterpoint to that is these are being dispensed with perhaps some false promises attached. Not every patient that receives remdesivir is going to see a 31% reduction in their length of stay. Not everyone who gets convalescent plasma is going to see a 37% reduction in severity of symptoms and recovery time. Now, if I give everyone with a headache a 500 milligram Tylenol, for most, for all intents and purposes, that Tylenol is going to work effectively because it has Tylenol. It has Tylenol in it at the measured concentration, and it is deployed to the patient with that measured concentration, so it's going to work. It's going to reduce pain and it's going to reduce fever. Convalescent plasma, we don't know yet.
Oh, I have a question about this.
Okay.
So I understand that you don't want to give something that isn't definitely proven to work. But out of all of the therapeutic things that we could give people, does this have the least downsides? I'm just thinking about this,
Sure.
the H word, and that this can get really complicated with people that might have cardiac issues, right?
Yes, correct.
Does plasma treatment have something that I would not know about that is risky to people?
For the most part, it has the lowest risk of side effects.
Okay.
But again, we can't say that is 100% true. There is a risk that the screening process and the preparation process is not uniform for everyone who's had plasma collected from them, and therefore we could potentially introduce other infectious agents along with it. We could also introduce something that could cause anaphylaxis in that patient, so they'll get a severe allergic response. There's definitely, we can't say there's zero risk, but it compared to just giving, antibiotics or anti-parasitics or anti-whatever, malarials that have known cardiac side effects, known whatever, convalescent plasma is somewhat safer. So that is a good question. Okay.
Could I ask a question real quick?
Yeah.
And I understand with the different strains, for convalescent plasma that would cover all strains.
Oh, Terry is now graduating to a molecular biologist thinking. He's thinking along the lines of a molecular biologist. Okay, so great catch. If you're pulling all of this stuff and you've anonymized it, you will not know whether you're getting plasma from a patient that got infected in California or plasma from a patient that got infected in New York. And it is not strain specific. So you could potentially get plasma from someone who suffered from a completely different strain that you're suffering from, or someone that got infected when that mutation that we talked about, D614G, was not prevalent, and so their plasma is totally not effective. Yeah, absolutely. Very good.
The thing that popped that into my head is, of course, there's another story in the news about someone who's reinfected with a different strain.
Yes.
So I'm thinking convalescent plasma and a different strain. How effective could that possibly be?
It wouldn't. It's exactly, you nailed it, because it would not be effective. What we're looking for, and I think it's a good segue to what I'm going to talk about next, is these are emergency solutions, right? As we have grown more sophisticated with treatment, we can set aside emergency treatments and more, do more precise treatment. And one of the things that Terry caught is that this is a scatter shot method. Okay, if you can use a rifle versus using a shotgun. And when you're giving convalescent plasma, it's a shotgun, right? These pellets are going to just go out like this into the patient's body. And what we're hoping for soon is that therapies come forward that are based in a molecular medicine world where we're targeting the virus, okay? And we're targeting the virus that's in the patient, not the, this whole population and subpopulation of strains that are out there. And if you give convalescent plasma from a patient that suffered from a different strain as the patient you're treating, it's it's a very shotgun method, and it's going to miss all, if not, the virus is just, those antibodies are not going to be useful.
Deborah has a good question in relation to this. So she's asking, when you talk about the efficacy of different doses of plasma, is that solely due to the number of antibodies, or do they differ in some other ways? Some more active against the virus than others?
Both. So what Terry mentioned is it's not just the concentration of antibody that's in the plasma, which is what I was talking about based on the dose, the inoculum and response. So severity of illness predicts the amount of antibody response in that particular plasma. And then the second thing is called specificity. So specificity is, are those antibodies that are in that plasma specifically going to go after the virus that's in that patient that you're trying to treat? And so that is going to be in question as well. Concentration and specificity of convalescent antibodies determine efficacy. Okay, so you need a good quantity, and they need to be specifically, sticky for the virus that you're trying to treat. Okay, and why? Why do they have to be sticky for the for the virus you're trying to treat, because this is what it looks like. Once you administer it, this is what is going to happen. Do I have that? Yeah, this is what's going to happen. So these are the antibodies that are in green, the Y-shaped antibodies, all right? What they're attempting to do is attach to that spike protein, the red protein. If they are highly specific, meaning lock and key, all right? So the lock and key is machined perfectly, the key fits perfectly in the lock, and boom, you get a nice tight fit. Energy of activation is nice and high. Energy of binding. So then the virus's ability to go and attach to these receptors on the human cell is defeated. So if you give a high concentration of highly effective specific antibodies, the virus cannot bind these receptors effectively, can't infect effectively, all right? So that's why you need to be more aware of what kind and quality of plasma that you're delivering to the patient. So how can we get around this? Well, I'll do this first. How can we get around this? Well, instead of collecting it from patients, we can engineer it, all right? So we can actually gather the antibodies that have the highest energy of binding and molecularly examine their sequences. So a protein consists of a string of amino acids. Those amino acids are coded for by a string of nucleic acids, so RNA or DNA. So for every 3 nucleic acids, you get 1 amino acid. It's the central dogma of molecular biology. And what we can do is sequence. Sequence is getting the entire, what is ATCG of the virus, the sequence. We know that the virus has 29,000 bases of RNA, and they code for about 41 different proteins. We can figure out what that spike protein is going to look like. Even the mutants, we can figure out what the mutants look like. And once we've figured out that, we can engineer, we can put that gene for that antibody into a bacteria and make that bacteria produce the protein in bulk. Okay, then you purify the antibody, and you can now have a highly specific therapeutic antibody, monoclonal, that you can deliver in a reliable, known dose to your patient. And so right now, these 2 companies are working on this, okay? Patient-derived monoclonal antibody, it is blah, 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 recovered, and then what you can do is we then can put the appropriate concentration, appropriate specificity into the patient via IV. Regeneron is working on this, monoclonal antibodies generated against the spike protein. And then we talked about llamas a while ago. Llamas can be infected. They don't get sick. And then we pull their antibodies, purify them, and you could use those. But there are lots of ways to engineer your own monoclonal therapy without having to pull it out of a patient that's recovered. Okay. And
yeah, question on FB that it goes towards all of this.
Sure.
Linda Menihard asked, is there a test now or in development to test the convalescent plasma for the exact strain of COVID?
Oh, yes. The answer to that is yes. However, can you see me, or am I sharing?
We see you.
Okay, good.
Right now the page isn't sharing.
Okay, so that's a very good question, but the level of processing required to get strain-specific categories for the plasma is pretty intense. So you need to pull, plasma, you need to sequence the protein, the actual antibody, and then match it to the strains, okay? To do that for each patient is very intensive. it requires quite a bit of work. So you'd have to do that with certain, the more efficient way to do it is with assumptions that you would not allow plasma from New York to be deployed in California. You wouldn't allow plasma in Louisiana to go up to North Dakota. You would just have community-based deployment, collection and deployment of that would ideally create the most effective source and target for the plasmas. Right now, I don't know if we're doing that, to be honest. I don't know if the FDA is mandating that we do that. Okay, so it is definitely possible to get an idea of what strain the patient was infected with. There's no doubt. In New Zealand, they're doing that. Every patient has their viral sequence done, and they're backtracking to see where that might have come from. It's not even, oh, who did you talk to? Where did you go? They're sequencing the virus on each patient. Why? Because they're infected. Their most recent outbreak was 24 people, not 40,000 a day. Try doing 40,000 viral sequences on plasma a day. No. Okay. So yeah, good question. Any other we should catch up on or keep going? Okay. So go back to this. All right. So we've had, where did this idea come from? We have had a therapy called intravenous immunoglobulin for a long time. And what we do to homogenize the effectiveness of the IgG is that you take it from 10 to 15,000 patients and you pool it, and then it's homogenized, and it's usually given to patients that have suppressed immune systems or some sort of antibody production deficiency, bone marrow suppression, and things like that. And this can prevent them from getting ill generally. So you're not taking IgG that is specific to a recent infection or disease like COVID, but you're just taking 10 to 15,000 healthy individuals who have regularly lived a normal life of getting sick and getting better from normal daily infections and common cold and flu and whatever, sore throats. And when you pull that all together, what you have is an effective pool of IgG that will help to neutralize common infections in patients that can't produce it themselves. Okay, so this IVIG is not specific to a disease. It's just to bolster the immune systems of people who can't produce this effectively for themselves. All right. So we know the exact dose, so 400 milligrams per kilogram of body weight. You can give it over a 3-week interval, and it does a lot of great things. So what it does, I'll go in order here. Hold on one more second. Sorry.
It's where John comes on and does this.
Bernie, you're killing me.
You've got the age, Karen. I've got the dancing fingers for John.
So, okay, so this is very technical, so I'll try to go fast. The Y-shaped antibody, which is hold on. So this is the Y-shaped antibody, right? That's Y. That is actually in 3 space, looks like this in 3D space, okay? And it is divided into the upper part of the Y and then the stick part of the Y, right? Okay, so this is floating around in your bloodstream in massive quantities, but it's so tiny that it's a tiny protein, but it does different things based on what part of the antibody we're talking about. So the upper part is the one that actually binds to viruses or binds to bacteria, and then that binding can do a number of things. It can neutralize the bad part of the infectious agent, like the spike protein or something that is used by the bacteria to cause damage to your cells. Or it can also sequester other parts of the immune system to come into the fight. Okay, so if it binds and then the bottom part comes along and binds to other parts of the immune system, like white cells, okay, then it activates a whole another level of immune response. And this is important because it's not just that, I mean, look, so when we talk about convalescent plasma, we're delivering plasma, which has a bunch of other stuff in it. We're delivering antibodies. So the majority of the active antibodies are a result of the most recent infection. So you got to gather it at the right time. You got to gather that at the peak of when those antibodies have been produced in response to the infection. If I take it too early, like, you know, Terry gets sick, recovers next week, and then I'm like, Terry, let's go. Got to get your plasma donated. Well, it's not ready yet. Okay. It's going to take 14 to 16 days for him to have the requisite amount of antibodies to be an effective donor. It's, you know, and then it peaks over to the next 2 to 3 weeks. And then it starts to kind of settle down into a memory status. That's what protects him from reinfection from that strain for the next couple of months, right? If I take it too early, there's not enough there. If I take it too late, then also I have a suboptimal amount of antibody, all right? So are we making sure that everybody we take convalescent plasma from right now is that plasma is being taken at an optimal time? What's your guess? No.
I'm going to say no. We can't even, we're not even collecting our data in a uniform way.
Yes, yes.
So I would make the assumption that we're not collecting our plasma in a uniform way.
Correct. So this is what frustrates people like me and others, that complex problems often require complex solutions. Simple problems can be solved simply, but you need to know what the difference is. If you assume a problem is simple, like we'll just give convalescent plasma to everybody, then the results are not going to be as expected, all right? The details of this are very detailed, for lack of a better word. So, you know, this is going around so this process here that I'm showing you of engineering the right amount of IgG for the very specific disease that you're trying to tackle, using a platform that's reliable and producing antibody that's specific and in the right concentration. This is ideally where we should be paying a lot of attention and trying to get this up and running. Good, very good, and we've got a good start here. This got us through with some patients and helped a lot of patients, it seems. We need to know more about it to say that it's truly effective. Otherwise, we're just pushing it, and it's not fair to everyone. Okay. All right, that's what I've got on convalescent plasma. Does anyone want me to cover anything else when it comes to therapeutics? Oh, okay. Well, yeah, I mean, that's largely been debunked. The MyPillow guy is, you know, been debunked and relegated to the trash heap. And I can't believe, looking back, the amount of effort that all of us put into having to refute. I just want to take a moment and give, just those slides should be an example of how much goes in to assessing efficacy and safety. Okay. Just because, like Terry put this beautifully a couple weeks ago, he's like, just because some guy in France gave it to 36 patients and then claimed they all recovered, and that found its way over the pond and into the ear of our president, and boom, off to the races.
29 million doses international reserve now. Check, please.
So yeah.
So for people that are new to the medical and science world, it is a common humans are fallible, right? And they want, in their mind, they want to be the ones that have found the cure.
Yes.
And this is something that happens even outside of the pandemic, and science knows that this happens, and that's why.
Yeah.
Because doctors can be biased towards their own assumptions or hypothesis, right?
Yeah, I think the drug I refer to as Avandia. Last town hall, or the one before that, where it was, I can't even remember what class of drug it is, and it was pulled from the market, I think 9 years after it was put on the market. Might be shorter period, maybe 6 years. But imagine that it went through all of those approval processes, and it went through all of those trials, and it went through all the post-trial data collection, and then it went through clinical usage for years before we discovered that it hurts people. Okay? Now we're in a pandemic, we're in the fog of war, and you need to be careful what you're pushing out there. That's all. That's all. Just be careful. Because the first law, I mean, what we adhere to as part of our oath is first do no harm. Okay? And inadvertently, many times we fail to first do no harm. And that is why medical errors are the leading cause of death in healthcare, okay, in healthcare delivery. It's not funny, but June and July are the worst times to get admitted to the hospital, especially teaching hospitals, because the fresh crop of interns have joined and they're handed the reins to patient care for the very first time in their lives. Okay? So what do they lack the most of? They don't lack knowledge. They've just been taught for the last 4 years the cutting edge of medical reference. You know, they've read the latest textbooks. They're exposed to the latest journals. They're exposed to all of the latest teaching material that guys like me are 20 years past. But what do we have that they don't? Experience. Okay? And when something new is pushed out into usage, what it doesn't have is the wisdom that comes with experience, the wisdom that comes with usage over a long period of time. So that's why I'm always cautious about anything that's new, and I'm imbued with that by my dad. You know, there has been a culture, there maybe continues to be a culture, less so now, but like David just said, you know, the drug companies about 25 years ago, they'd show up with a big lunch and all kinds of shiny pens and tickets to this game or that game. And all you had to do is prescribe their newest, shiniest med, okay, that just came out. And imagine all the specialties, pulmonology, nephrology, cardiology, you know, all the way down the line, up and down, primary care. All these guys and girls show up looking awesome. They're all young and gorgeous, and they get you great food, and they give you tickets to this and that. And all you have to do is prescribe this new shiny thing. Kills people. You know, kills people. First, if you don't know how to use it, if you don't use it right on the right patients, if you don't use it at the right time, there's so many factors that go into therapeutics, and only experience teaches you that. So, yeah. How we doing on Facebook? Anyone?
No, I did want to say that, you know, you were talking along those lines, and the way this has evolved into our country's consciousness is sort of like, people seem to think that, say the H word, you know, it's their right to try and take that
Yeah.
since it's put out in the public specter, and it's their choice to make. They will chase their doctor down and hound them to get them, you know, I want this new miracle drug, it's my right, you know. And a lot of that is happening in this country right now. Uh, they said in the last week 5 people have been treated for ingesting bleach in Texas.
It's yeah, and you and I know this oleandrin thing. It's going to be a disaster. People are, as soon as they can find a way to get it, it's something horrible is going to happen.
It's poisonous enough to kill a small child.
Yeah.
And it could put a hurt on an adult.
So,
let alone whatever permanent damage it can do to your innards.
yeah. So let's quickly cover, because now I do get people messaging me and saying either they've been exposed or they are awaiting their test result and they have runny nose, sore throat, or something. They're early on. What do I do if I'm waiting for a test result, if I'm asymptomatic? Well, what we recommend obviously is control your fever, monitor your fever at least 3 times a day. Stay hydrated. A multivitamin is recommended. Make sure that you are well nourished, and that means vitamin D and vitamin C are highly recommended because of their effects in the immune system and the immune response to viral infection. You would do the same thing if you had the flu. I would recommend vitamin C. Fever control, hydration, rest. Okay. Then if things get to the point where, yeah, I'm positive, I know I've got more than a sore throat, or I've got more than, I've got more than a runny nose and a cough, I'm starting to get sick. I feel like a high fever and things like that. There's not just the Tylenol. Hold on, guys, 1 second. It's loud. Little quieter, please. Yeah, so the recommendation is that you do all the things that you would normally do for flu, but once you start getting that chest tightness or an intractable fever or severe malaise, which is now like a signal for attention by healthcare team, then, and I think I have one friend right now that's already reporting all of those things on Facebook. So I'll be checking in with her also. And those things are indications that you need to go see a doctor, okay? Call ahead, make sure that whoever knows that you're coming and has the appropriate preparation time, because obviously in this era, all of those healthcare providers should at this point be ready to handle someone who has COVID, but it's also good for you to call ahead and let them know, okay? Should I take anything else? Well, I sincerely don't recommend anything else, okay? Control of fever, hydration, daily multivitamin. If you feel ill, if you start to feel worse, then there's other stuff that the healthcare team is going to try to use, okay? Once you show that you need further stabilization, further treatment. If you start having high-grade fever, difficulty breathing, severe malaise, then they will start, and a lot of people ask, like, Karen knows, like, what was it, maybe 5-6 weeks ago, people were asking, what should I tell my healthcare team in case I get sick? At this point, I can confidently say that these protocols have been adequately disseminated. Now, I can't speak for every community hospital in every corner of the country, but most modern equipped hospitals with an internet connection should know that at this point we treat with remdesivir if it's available. If it's not, then definitely with the low dose, low molecular weight heparin and dexamethasone that we talked about earlier. Okay, those are going to largely control symptomatology and largely allow the body to do the work of healing itself. And they may add adjuvants. Okay, and those adjuvants could be, vitamin C, vitamin D. There have been tests, trials on the use of famotidine and other agents. And those other agents are, either going to be generally used only in a university setting, research hospital, big, clinical research centers like Mayo, Cleveland Clinic, UPMC, Mount Sinai, UCSF, Penn Medicine, all these other big centers are testing these other drugs that are immune modulators like leronlimab, tocilizumab, and others. And more information on interferon is coming through, and I expect within the next few weeks, interferon is going to be a core therapeutic. Okay. And you will be too sick to know whether these are things that are people are doing for you. So we don't need to cover these. But this is how we have evolved the protocol for the severe COVID patients to be handled. And proud to say my healthcare army all over the country and most parts of the world have been able to cut mortality even without a magic bullet by just being smart clinicians and utilizing these. Okay. So questions so far? Oh, we have a big crowd this week.
Let me look.
Nobody wanted to watch the RNC? Makes my heart warm. You guys wanted to join me and not watch your favorites on the RNC?
So, I can go on what I saw last night, but first of all,
wait for the post show. It'll be
yeah.
fun.
First of all, Chai had a question about vitamin D that I didn't see here.
Sure, go ahead.
So she asked, how often can you take 5,000 IU of vitamin D capsule? Right, so she's asking about dosage.
Yeah, 5,000 IU, so 2,000 IU. What did we say? I can't remember exactly now. I'd have to look it up. But whatever the US RDA is for the daily dose of vitamin D, double it, and you're fine. Don't go over double. Don't go over double.
And on zinc, what would?
Again, US RDA.
Can you double it?
Yeah, of course. Yeah, absolutely.
Okay.
You don't want to take any of these indefinitely at that level. Amanda Emerson is one of our friends on the page, and she has done research on why you can't take these things. It's a pandemic, I'm going to take it until the pandemic is over. No, you can harm yourself by taking too much of these minerals, copper and manganese and zinc. They can accumulate and they can cause problems. So, there's a lot of people out there, pushing the theories that these are, they have outstanding antiviral effects. And if you take it with this drug, it pushes it inside the cell and makes it even more effective. Well, just.
No, it ain't UV light, but hey.
So yeah, just take, supplement your normal diet. You actually do get a lot of trace minerals and a lot of these things in your normal diet. Just in this era, it's best to take a little more than usual.
I got a statement and a question on Facebook. One, Linda Minihart said daily dose of vitamin D is only 400 mg. Many physicians prescribe well over 10,000 mg of vitamin D.
No, they could probably, they're prescribing 10,000 international units. Okay, and that's different from micrograms or milligrams.
And
I don't know what mg or mcg.
she said MG,
Mil,
especially in
mil,
patients who are deficient or have MS.
yes, correct. You can give large vitamin D doses in those patients, and that will be in international units. Not.
Then there was a question from April Diana Niver who asked your thoughts on the preliminary studies that people may be asymptomatic due to resistance from other coronavirus exposures. Didn't we talk about that at one time?
Yeah, molecular mimicry. That's absolutely a possibility. Like we even saw a paper that's being followed up by other researchers that those who have received MMR or the tuberculosis vaccination, the old vaccination, um, that those patients have seen lower symptomatology and lower severity of COVID infection. And we talked about how the immune system, essentially with antibodies, is a lock and key system. But remember that at scale, one lock, one key may fit multiple locks. Okay. You can't ensure that that key is specific only to that one lock. Eventually, as a result of manufacturing error or some small molecular errors, those antibodies will be effective against other things. Okay, so if you have exposure to something as a result of vaccination or infection, and those antibodies that were produced have some sort of molecular mimicry um, in terms of their morphology, their shape and three space, then they can be effective against other infectious agents. Not optimally, but good enough to provide some protection. Okay.
We're up to date on that.
We're good? All right. So let me just, uh-oh.
I didn't do it this time.
Where did you guys go? Zoom, here you are. Okay. Share screen. All right, play from current slide. All right, so anything else? There are some people who are recovering that are struggling with recovery. We call them long haulers, and we will continue the same kinds of therapy with them. I don't know if we have anybody listening that has recovered from COVID. We'd love to hear your story. We'll open up in just a few minutes now for the last half hour to go from this panel view to the general, everyone being able to talk. And if you have any stories to relate about recovery or illness, we'd love to hear from you. But yeah, we're using the same stuff to treat these patients afterwards by giving them low dose instead of the high dose, short duration dexamethasone. We give them a low dose corticosteroid to keep their immune system calm as they recover and prevent it from doing any further damage to already fragile pulmonary architecture or vascular architecture that has been damaged by the viral infection. Other anti-inflammatories that are being tested are statins, aspirin, melatonin. Of course, keep going on the supplementation, vitamin C and vitamin D, things like that. Okay. Yeah. So interferon beta is a rising star in the treatment of COVID, and so is vasoactive intestinal peptide, which has been engineered, synthesized as a drug called aviptadil. And so these are now undergoing trial. Now there's a COVID tracker for therapeutics. It is much more reliable than our White House, and you will be able to actually see what stage of the process of evaluating efficacy for each of these and who's conducting it where. And so the therapeutics tracker is right there. And this goes on our resource page so that you can look this up and use it for your own purposes. Yeah. Any questions on any of these drugs? Or should I just move on to vaccines?
Let's talk about vaccines.
Okay. So vaccines, again, it's unfortunate that we have to interject a little bit of the political environment that we're in right now. We have an election coming up in this country, as you all know, and as a result, the party in power is doing its best to make the case that they've done their utmost and continuing to do their utmost to handle what's been ravaging the country for the last 6 months, which is this pandemic. The numbers tend to speak for themselves. We've lost the most citizens of any country in the world. We have the most cases of any country in the world. And there will be a tendency to politically try to cover themselves by saying that we have a vaccine development that is unprecedented and in its speed. There is a et cetera, et cetera. And, to a certain extent, I will give credit and thanks to those in government that have pumped out nearly $6 billion to the private industry to utilize for development of both vaccines and therapies, and to get them out into the hands of clinicians. However, knowing what I know, which is not always a blessing, certain vaccines are known to be reliable platforms that we've used in the past and that have been effective for millions of people for decades. Unfortunately, the one that has been selected to be fast-tracked is not one of those platforms. The one that has been selected to be fast-tracked is a novel platform that uses only the nucleic acid that has been identified, sequenced, and purified as what we call an mRNA vaccine. And that is what is likely going to be most likely going to come to market the fastest. Now, that's not because it is politically expedient for that to be done. It is going to come to market the fastest because it is the simplest platform to develop. Okay? So from a chemical biology, molecular medicine perspective, there is no simpler vaccine platform than mRNA. And so that is why the one that Moderna is using is going to race through all of these steps faster than the ones that have we been using traditionally, like live attenuated virus or inactive killed virus, or the recombinant version of vaccination. Okay, so some of the examples are, for inactivated is rabies vaccine. For recombinant is the human papilloma virus. We've used live virus, but attenuated so it doesn't cause the disease that's associated with it, with measles and mumps. We've used it, in fact, even with influenza. So live virus, but it doesn't cause all of the disease associated with the wild type that people get that causes disease. So these are much harder to produce and test. And there's a lot more chance of side effects or unintended effects when you're dealing with live virus, attenuated virus, or something that has been bioengineered. So they are going to take longer. Fortunately, the AstraZeneca platform, which has been spearheaded by Oxford University and the National Health Service in the UK, is neck and neck, maybe, with Moderna, simply because this platform was available as a result of all the work that went into it 12 years ago with SARS-1, SARS-CoV-1. So utilizing that, they were able to accelerate this version, which is we've described it here before as a sheep in wolf's clothing. So it's an adenovirus that normally infects chimpanzees, that is the right size and has the right appearance to look like SARS-CoV-2 to the human body once it's circulating in the endothelium of the patient, epithelium of the respiratory system. So it looks a lot like the virus that causes COVID, but it's not. And so it trains the body to protect itself from COVID. It's not ethical for me to make a recommendation on these two platforms when neither of them have been deployed and we have no data on them. But I just want to give you the background. The background is, one is a proven platform that we've used with lots of diseases, and we've succeeded over decades in preventing illness. One is brand new and has never been used before. Okay. So let's talk about questions.
So there is a question about symptoms from Katie. She's asking if you relapse. So if you feel better and then you go back to feeling bad, are you still contagious? I think they give a guideline. You start counting like from the day of first no symptoms, I thought, right?
Now that's a complicated question. Are you contagious? You're contagious from day 4 post-exposure through day 14. Now, between day 4 and day 14, you can actually not experience any symptoms, or you can actually get sick and experience symptoms. 80% of the people who get sick will have a very mild course of disease. Let me break this down. 40% of those exposed will not feel like they've gotten sick, and they can be contagious from day 4 after exposure up to day 14. They'll be shedding, just like somebody who was sick during that same period of time. Let's take the other 60% of people. Of those 60% of people, 80% of those people will get through this without much problem. Okay? It will feel crappy and they'll feel sick for 4 to 8 days, and they'll get through it no problem. About 12% will get the worst flu of their life, okay? And they'll really feel sick. And that 12% may actually not even seek healthcare treatment, from a healthcare provider, but they survive it at home, and they're like, oh, that was the worst flu of my life. Then about 6%, 6.5% to 8% will require hospitalization. Okay. And then we still see that 2.5% may require ICU level of care, 2% about ICU level of care. And of that 2%, about currently today, 0.5% will die. So that's how it breaks down. Now, if you had asked us these statistics in April, I think we covered this, that it was little higher in terms of the people that go into the ICU, and it was around 3 to 4%, I think we said. And then the people who died was around 2%, as high as 2.5 during the peak in New York. So we've cut down mortality significantly. We've cut down trips to the ICU significantly. And we have been able to treat the patients that get really sick and have to go to the hospital. In the hospital, we've treated them more efficiently in the last 3 months.
But when you're talking about long haulers, you're talking about, you're not talking about people that have the virus you're talking about symptoms that are lingering from the effects of the virus.
Exactly.
Okay.
Exactly. And long haulers are about 30% of that 12% that have had the worst flu of their life. Got it? So, I'll break this down for us next week, because I'll just put like 1,000 people and break it up into the people that out of that 1,000 that are going to go into those categories. But, we said 12% have the worst flu of their life. 30% of those people that have the worst flu of their life are going to have some sort of lingering symptoms, which we call morbidity, for months afterwards. 50% sorry, 50% of that 12%, 50% of that group will have cardiac and pulmonary injury visible on testing post-recovery.
I was just going to say that Katie is saying that she believes she's a long hauler, and she was told that if you have a fever, you're contagious.
No, that's not true. That's not true at all. Who told her that? Anyway, I don't want to know.
I don't know. We'll find out in the after show. We'll ask her.
So we are now at the stage before the after show where we can invite attendees to be panelists. Do we know how to do that?
I think I do.
Okay, yes. So let's bring Katie in. Whoever else wants to join during this phase and share with us, just raise your hand or tell Terry and Karen, and they'll add you.
All right, so we're officially ending.
Yes. We're not going to post show yet, so we're still not political.
Oh, okay.
We have to answer these questions, and then we can go to post.
We're still on Facebook Live.
Yes.
Okay.
Yes,
that's
we
fine.
are for another half hour, I think.
Yeah,
so do not confess any crimes right now, everyone.
No. Okay, let's go to gallery view. All right, here we go. And Giselle's with us. That's awesome. Don's joined us again. Lorene's here. Catherine, good to see you guys. Is Katie there? Yes. David. Awesome. If Katie wants to share her story, that would be great.
I got a couple of questions here. One was, Lorene said in the chat here, will there be different vaccines used for different age groups, like
Yes.
the one for young people, one for 20 to 65?
Yes.
And also, Linda on Facebook said the subset of the population are anti-vaccine, and now I'm seeing on social media platforms are stating they will refuse any COVID vaccine that comes to market.
Mm-hmm.
How do we overcome this with a successful vaccine?
Good question. I don't think we will overcome their reticence to taking the vaccine through any, I think that what has happened recently, which is really somewhat of a tragedy, is there's been such a severe undermining of, and we talked to JJ about this, of faith and trust in public health recommendations. Some believing that they're politically motivated, some believing that they are attached to some sort of conspiracy against the U.S. public, or whatever. And then there's those that have foregone all of that and just have a severe aversion to vaccination as a as a principle of public health, that continue to cling to some evidence, whether true evidence or not, that or claims that that will cause some other disease. So if I give this to my kid, or if I take this, I'm going to end up with something else. I'd rather not take this vaccine. Okay, but then there's those of us that have to live in the world, and those of us have to live and function in the world, um, and go out and work among others, should see that there is not only a benefit to ourselves, but a benefit to society as a whole for us to get vaccinated. So it's the same as 70% or 80% of us not being anti-mask, who are willingly participating in distancing and mask usage. So if we get the same number of people that are willing to put a mask on and be responsible about that, that same number of people go and get vaccinated when a safe, effective vaccine becomes available. Not to sound callous, I'm not that worried about those that are refusing it. I would actually like David to comment on this, because he probably has some insights that I'm missing.
You are 100% correct. You asked before, how do we overcome this? And I'd say we don't.
Yeah.
We don't. And we prove that it works. We focus on those who we can influence. We focus on those who are in the middle, people who are not swayed either way. And if you're familiar with Dan Kahn's work, I actually just shared a quote to the chat right now, or I almost did. He finds that the deposition to construe evidence in identity congruent rather than truth congruent ways is a state of disorientation that is pretty symmetric across the political spectrum. And interestingly enough, sometimes even those who are extremely scientifically competent can actually fall into those same categories, whereas those who are scientifically curious are more flexible across a number of different metrics. So I don't have that right in front of me. I was actually searching for that as we were talking about it. But how do we overcome that? I don't think we do, and I don't think we waste our energy on that. I think we focus on people who are open to it.
I totally agree. I think that you just build faith with performance. So if we have something that is effective and that protects our kids, and they go back to school next year and they're vaccinated, and we've seen low COVID transmission rates across a huge spectrum of society that's gotten vaccinated, it's going to speak for itself. Between now and then, it's a waste of breath. So it's a waste of effort, like you said. Now, can we ask Katie to join and have her tell her story? Because that's probably important to hear.
I can unmute her. Let me see if that works.
Okay, can you hear me?
Yeah, Katie, I can hear you. Go ahead.
Okay, so you want me to start from the beginning, or is that
Sure.
Okay, so I got sick in March and tested positive. I thought it was a sinus infection at first. So at first I didn't, get tested immediately. And then, fever started, and which, isn't normal for me. So I called my doctor, and at first he didn't have me get tested, which I thought was strange. Then I went and got tested, and it was positive.
Katie, where are you located?
I'm in Shavertown.
Oh, my neighbor. Okay, keep going.
Oh, yeah. So let me see where. made me lose my train of thought, which is hard these days. So, let me think. So, I couldn't breathe. That was the worst of it for me. It was terrible. I should have gone to the hospital. Like, I absolutely should have. But I'm stubborn, and honestly, I didn't want to go on a ventilator. So I suffered at home. But I was also in touch with my doctor. And we had a plan for me to stay home as long as I possibly could. I did lie about some numbers because like I said, I refused to go. I just absolutely refused. So, but I made it through. I did have his care. So it was off hours, and, he was in his full PPE and everything. And, but it's been absolutely terrifying. I'm still sick, I tested positive for 4.5 months. We were trying to get my boyfriend who lives with me to go back to work. So I kept trying to test to get, to be cleared. And so, which didn't happen. But, and now I'm still dealing with everything. I'm having daily fevers, 101 to 104, memory loss, brain fog, my hair's falling out, still shortness of breath, chest tightness, trying to think. There's a bunch of other stuff, but that's the main stuff.
Have you been prescribed anything? Are you taking anything for these symptoms?
I do the inhaler. I have 2 inhalers, Buterol, and I can never remember the other one. Starts with an S.
Okay.
I've done all the steroids. I'm on vitamin D. A lot of vitamins.
And you said this is your 4th or 5th month of illness with fever still?
This is actually, yeah.
Now, do you work in healthcare?
Nope.
Do you work prior to this illness? Did you work around a lot of people?
No.
Do you have any idea where you got this from?
No, I do not.
Okay. And your boyfriend lives with you, has not been ill at all during this time?
He actually just got sick the last few weeks.
Okay.
And his test actually just came back negative.
All right. And have all of your tests been done at the same place?
No.
They've all been positive?
1 negative in between.
Wow.
Wow.
Yeah, your story is not unique. It's horrifying, and I'm sorry you're going through it. Yeah, there's a lot of people in this community, called long haulers, that have suffered for months like you are right now. Again, I'm really, really sorry to hear you're going through this. Has there been a period of time that you could describe as the worst of it, and then this being where on that spectrum?
I would say it's been kind of crazy. March was the worst, March and April. And then there would be times when, a day or two I felt better, and then I would get worse again. And now is really bad too, but in a different way. I'm extremely tired, like to the point where I can't keep my eyes open. And I'll sleep for like 4 or 5 hours. And people can't wake me up. And it's awful, you know, and just walking, I can't breathe, and that's not me, you know.
Yeah.
And I don't want to live this way, you know. I never did before, and it's awful.
Yeah. Okay. Well, I mean, after this and offline, I'm going to see if there's something I can do to help. But yeah, thank you for telling us about it. And I just, I mean, I'm struggling with it too, not being sick, just struggling hearing everybody's suffering and trying to figure out if there's resources that I can gather for these long haulers, because and for you, that haven't found any relief or recovery. And it's just extremely physically debilitating, but I'm just talking to people. It's mentally, just, it really wears you out. And so outside of this community of doctors or whatever, have you reached out to anybody like sorry guys, sorry, at the like at a unit, like down at Penn Medicine or outside to get like a higher level of investigation?
I haven't yet, but I plan to.
Yeah.
I plan to. I'm actually going to call my one doctor tomorrow as a urologist out of the area. So
Yeah, that would be a good idea. When you've started to exhaust, what your local providers know or can do for you, it's definitely time to look towards other investigators that might know a bit more about what you're going through, or have other patients like you that are experiencing something that they can provide.
Yeah.
Yeah, thank you for sharing your story with us.
Yeah, I'll be in touch, Katie, I promise.
Glad you have this. This is nice.
You're welcome. You're welcome. Did David go away? No, there he is.
Nope, I'm still here.
So any news on your end? Any new discoveries? Anything you should bring to our attention?
No, honestly, I'm hiding in the background. I don't have much to contribute, honestly.
Okay, that's fine. That's fine. What about anybody else?
Madeline still has a question about the vaccine developed by the group at Baylor Medical Center.
Yeah. Baylor has an outstanding platform, and actually have gone through the animal models. And so I think his name is Dr. Hotez at Baylor. He's gotten really loud actually in the community. And he's gotten kind of political too, which is fine. Not saying anything about the response. And I think, he's been driving he's been part of the group of investigators at Baylor that are driving Baylor's vaccine. I don't know how far along it is. One thing I know, it was ready for commercialization for CoV-1, for SARS-CoV-1, and it just, nobody picked it up. But I haven't heard, and I've kept my eye out for any further press releases or anything, any news out of their group, anything from Baylor, and I haven't heard anything. There's nothing pre-press from them either. Right now, the vaccine pursuit is dominated by AstraZeneca, Sanofi, Johnson & Johnson, and Moderna. And the one thing that I wish was not happening was that any of these companies were being favored, because it also taints the science news that comes out about them. When one group is favored, everybody's attention goes to, what's the next press release for Moderna? What's their next thing? And then everybody else that's doing good work, we don't hear from them. So that's also problematic.
I call that stock manipulation.
Yeah, right. Yeah. but there's a steady stream of news out of AstraZeneca about their progress. Oh my goodness, these guys. One sec. Talk among yourselves.
We are all his dog right now, by the way.
Yeah.
It's like an I'm Spartacus moment. Every time that dog goes off, I'm just like, yeah, mess shit up.
I come on here and do the John dance.
Oh,
Since I'm
absolutely.
not here, yeah. There you go. There's definitely a John Dawes maneuver right here.
Terry, you got points on the board tonight with your, was it your cynicism, or what did you get points before for?
I always get points for my cynicism.
He asked a scientific-y kind of
Oh,
a
yes, that's
question.
right. You became the primary investigator here. I forgot now.
Yeah.
It-
Boy
it
Scout
was-
virologist.
It wasn't just as pithy observations. It was deep.
And what did I miss?
Oh, we were just talking
how-
about Terry's good question.
Yes. Yes.
A good question for the week.
Should we
Yeah.
end the Facebook Live and go to our?
Yes. Yeah,
Okay.
I'm ready to go. Yeah, good to go. Everybody's got questions done. Nothing we want to let out to the public.
I think we're all caught up.
Okay,
everyone.
good, great. Well,
Yeah, so I
without-
know that John sent me some instructions.
all right, let's see if you can faithfully execute on John's instructions.
Okay, I'm going to close it with everyone here on the frame. How's that?
Yeah.
It was for our 23rd week with Dr. Sanjay Udoshi in our COVID town hall. I thank everyone for joining us on Facebook and on Zoom. And we'll be here next week and we'll do it again. Thank you very much.
Thank you, everyone. See you next week for the last one of these. See ya.